Good morning, all of you.
Ma'am,
So we were discussing advanced diagnostic aids. This is part 2. I'm taking. Last time we have discussed advanced diagnostic aids which are there in clinical diagnosis. Okay, why there is need for advanced diagnostic? Because periodontitis patient comes frequently to you for maintenance purpose. Got it? So frequently patient returns back. So you have to assess whether your treatment is working or not. How you are going to assess that on the basis of your clinical feature. I am audible to all of you.
yes, ma'am.
Huh. So on the basis of clinical features, the pro forma which you use while recording case history. So periodontal status clinically you check. For diagnosis of periodontitis, you check periodontal probing pocket depth. Then you check mobility of that particular tooth. You check clinical attachment loss, right? And when you treat, you remove plaque, calculus, and again patient comes. So how you are going to evaluate? Again, you are going to reevaluate by using periodontal probe. You will probe. You will check probing pocket depth, whether it is reduced or not. If it is reduced, your treatment is working it properly. If periodontal probing depth is not reduced and it is increasing, means what? Disease is worsening. So for this purpose, to evaluate every patient, when you write in your case history treatment plan, after every phase, you write maintenance phase, right? So maintenance phase from periodontal point of view is very important, and this maintenance phase only you will able to evaluate when you check probing pocket depth clinically, clinical attachment loss, and that whatever you have checked at first visit, it should be checked again at second visit. So while checking reproducibility of that particular aid is very important. Reproducibility means what? In the same area, same point, and in same angulation, same direction, you should insert your probe. If you are checking your pocket in the mesial side of a particular molar at first visit, in the second visit again at the same point you should insert your probe, in the same angulation you should insert your probe, then and then only you will able to measure periodontal probing pocket depth correctly. Got it? So for that purpose, reproducibility is very important, which we have discussed in our previous part also. Got it? So reproducibility. Who's who has not muted? Check it out. Who has not muted? All of you mute your device, phone. Okay. So what we were talking about, that reproducibility of that particular instrument or diagnostic aid is very important. For that purpose, you should maintain proper position of that patient, proper probing force for clinical purpose, proper angulation. It should be parallel to the long axis of the tooth, and it should be guided by a stent. Stent is nothing but it is prepared with the help of cold cure acrylic, and you will mark groove, so that from that groove only you will able to insert probe in your that particular area. So probing depth is measured properly by using periodontal probe. Got it? So clinically, how you measure for that advancement in periodontal probe, we have seen in the last class. We have seen that manual or first generation probe followed by second generation probe. Second generation probes are nothing but pressure sensitive probe. Then third generation probe, which is computer assisted probe. Okay, so that are the advancement which are taken place in clinical diagnosis. that is using your probe. Okay, so radiographically also advancement has taken place. Basically, for diagnosis purpose of a periodontal patient or periodontitis can be diagnosed clinically, secondly radiographically, third you will able to diagnose by identifying microorganisms which are associated with periodontitis, and fourthly you will able to check the molecular with the help of molecular test or enzymatic test which are released in periodontitis. Okay, so these are the four different ways by which we discuss or we diagnose a particular case as a periodontitis. So, for that purpose of diagnosis in advancement, first clinical advancement or clinical diagnostic aids, advancement in clinical diagnostic aids which we have seen in the probing. Secondly, advancement in radiographic diagnosis. So in radiographic diagnostic, there are different ways you people have seen in conservative department. So radiograph, how in conservative department it is taken by with the help of RVG. You people know it very well, I think. Silicon plate is there, small that plate is there which is kept particularly properly in that particular position, and then we take a radiograph. So this is the way or this is the advancement which has taken place in radiographic diagnosis. But few more advances which has taken place is subtraction radiography. Okay, digital subtraction radiography or subtraction radiography. Subtraction means what? Subtraction means minus. We know that this term is mathematical term. So subtraction radiography means what? Subtracting the unchanged part from radiographs. Means unchanged anatomical structure which are there. Means suppose a tooth is there which is not going to change anymore. So tooth position or tooth is not going to change. Only the defect, bone defect, which has seen periodontal bony defect, if it is 3-wall, 2-wall, or whatever kind of radiographic bony defect. If you are going to use bone graft, you are going to regenerate that particular area while doing surgical phase of your treatment plan. Then you have to assess. See, these radiographic assessment are useful when you are going to regenerate a particular area, regeneration of a periodontal defect. If there is 2-wall bony defect is there or 3-wall bony defect is there, then you are going to stuff the defect. During surgical procedure, you will open that periodontal area and you will feel with the help of a bone graft in that particular region. So that bone graft, you need to evaluate whether your bone graft has been taken, whether your bone graft is there in that particular region properly, and bone regeneration is occurring or not. For that purpose, you need to take radiograph in proper position. Every time you have to maintain the patient's position, proper positioning device should be there. So for that purpose, mainly long cone paralleling technique is very useful, and dissecting angle technique, that is also important, and proper positioning device which is there. That positioning device is very important. That positioning device is going to help to maintain the patient's position. So it extends. It is an extension arm which comes from the X-ray tube inside the oral cavity, inside the patient's oral cavity, where you attach your radiograph or plate in that particular region and ask the patient to bite. The bite should be done on a rubber-based impression part or rubber-based impression material, and the same bite you can use as a guide for your next radiograph. When you are going to take next radiograph, same bite record is useful and the same extension arm, so that that bite record will help you for recording the next radiograph also. So radiographs can be taken sequentially at every visit, every visit. So for first visit you take a radiograph, second visit again you take by maintaining proper position. So these 2-3 sequential radiographs are superimposed over each other. Superimposed means kept over each other like that. In a software, in a computer assisted software, you can place these radiographs superimposed position and unchanged anatomic structure like tooth or other unchanged structure can be removed, and you will able to find at the grayscale image, means whether the bone formation has occurred. Then you will see more radiolucency. In that, radiolucency is detected as a defect and radio opacity is formation, so you will able to find whether that bone regeneration has taken place or not. So you have to sequentially arrange radiograph over each other and subtract the unchanged anatomical structure, and you will find whether bone regeneration has occurred or not on that particular radiograph. That is subtraction radiography or digital subtraction radiography. Another technique which is there in radiograph is CADIA, that is computer assisted densitometric analysis. Densitometric analysis means you are going to check the density of that particular bone, or on the basis of that density the radiograph is kept on a screen and light is passed through that particular radiograph, and it gives you impact, or it gives you particular defect with whether regeneration is taking place. For that purpose also you need to arrange sequential radiographs and which is superimposed over each other, and then you will find whether this bone formation has taken place or not. Other radiographic advancement which are taking place is guided CT, local CT is there. Other than that, CBCT is there. But all these things, for that purpose, proper positioning should be there. You have to follow the proper positioning, and then only you can maintain that particular radiographic records, or it is going to help you as a aid. Now today we are going to discuss about advanced diagnostic aids which are taking place in third type of aid, that is microbiological analysis. So microbiologically we detect periodontitis um, on the basis of identification of a microorganism, because periodontal pathogens are there which are present in the sulcus, which are present in the oral cavity, and we can help that we can identify that periodontal pathogen whether virulent pathogens are present, and the number of quantity of that periodontal pathogen, if it is more, then disease is in active phase. And if it has reduced down, or if you will not finding the next visit, when next time again patient comes, you take a plaque sample and you again go for culture. That time, if quantitatively these periodontal pathogens they are reduced in number, then you will find that your treatment is working. So this is also a diagnostic aid. Microbiological analysis can be done with the help of culturing, direct microscopy. You can find microorganisms through direct microscopy, and immunodiagnostic methods are also there. Diagnostic assay based on molecular biology, as I have told you, and the other enzymes, host-derived enzymes are also there, which we can find. Uh, Other than that, means these enzymes are nothing but the proteins which are released when cell death takes place. Sometimes cell death takes place, or sometimes cell may release some enzyme. So these are the host-derived enzymes. Some enzymes, inflammatory mediators like cytokines and other products, then tissue breakdown products. Okay, so we are going to discuss today about this. So, microbiological analysis, for this, you need to have culture media. Okay, so for that purpose, you should be supported with a proper lab. Lab should be there. These techniques are very technically demanding and very costly techniques are there. Culture technique basically. For culture, another drawback of this technique or microbiologic analysis technique is what? Because periodontitis is not affected by a single periodontal pathogen. Because there are multiple pathogens, you need to identify finding multiple microorganisms is very difficult uh, from a patient. So, n number of pathogens are there, and that too, not a single pathogen is positive factor for detection of periodontitis. So, you need to find different species, around 1,000 different species are there which are colonized in the oral cavity, and almost these species are quantitatively 10 raised to 4, 10 raised to 5, or 10 raised to 8 times present in the oral cavity. So, periodontal pathogens like Actinobacillus A, PG, Canareella forsythia, Treponema denticola, Fusobacterium species, all these species are there which are periodontally pathogenic. So these, for that purpose, you need to have a culture technique. Culture routinely, two culture media are there, that is blood agar, agar, culture medium is there, McConkey's agar. These kind of culture techniques are there. But for this detection of culture technique, the main drawback of this culture is there should be microorganisms should be viable. Then only it will detect, then only colonies will form on the culture media. But most of these are anaerobic species. So when we take a sample from plaque sample from sulcus, and when it comes out, or we can collect the sample through the help of absorbent paper, small strips are there, or other than absorbent paper strip. In root canal, we use that paper point, absorbent paper points are there. That points can be also inserted in the sulcus, and we can collect that, and we can use it for culture. Okay. Other than that, we can use saliva also. We can use supragingival plaque also for culture. We can, but for periodontitis, mainly we should use subgingival plaque. Only problem is with culture media, some organisms require selective type of media, particularly selective media are there. Then other than that, uh, other than that, live microorganisms should be there. Viable microorganisms should be there. That is the main drawback. Here, the bacterial anaerobic culture, you can find that black spot, Priotilla intermedia, or P. gingivalis, yellow color colonies are there. Fusobacterium is there. So this is one image which is showing different kinds. This is blood agar culture media, and you can find different type of colonies which are present. So another technique by which we can find, we know now disadvantages of culture media. Okay, so it is very costly. Then another is it requires viable microorganisms. Other than that, technically demanding. Uh, all these things are there. So because of that, again, time consuming is there. Time it requires for colonies which grow. It takes time, but it is a gold standard method by which you know quantitatively as well as qualitatively. Okay, qualitatively means which kind of microorganisms are there. That is qualitative assessment. Then direct microscopy, in that case contrast microscopy can be there, or dark field microscopy is there, to which motile microorganisms, mainly motile microorganisms, can be detected with the help of motility, means the microorganism which move, um, like rods, or mainly Treponema species, we can find with the help of dark field microscopy. But not all kind of microorganisms can be detected with the help of this direct microscopy. So this technique is also not good. Then immunodiagnostic methods. So all of you know that antibody antigen reaction, that is immunological reaction which takes place in the body as well as in the microorganism. All living things, they have antigen antibody interactions. So microorganisms, if all living human beings, they show this immunological reaction or immunochemical methods are there. So for that purpose, direct and indirect immunoassays are there, immunofluorescent assays are there. So these immunofluorescent assays, what happens? Monoclonal or polyclonal antibodies are conjugated with fluorescein marker that bind with bacterial antigen, and it forms a fluorescent. That color complex will be seen, and you can find out whether that microorganism is present or not. Other than that, indirect immunofluorescent assay, in that this primary complex which is formed is again conjugated with secondary antibody, and then this first complex will act as a primary antigen or primary antibody. like that. So secondary again it conjugates and it forms a color coded complex. So flow cytometry is there. One technique is there in which the lamellar or a single flow is. Am I audible to all of you?
Yes, ma'am.
Ha. Okay. So we'll continue. I think it is continued, no? 10 minutes. I think I can continue with this. So next is enzyme. Hello, Rahul. Rahul, I am audible.
Yes, ma'am.
Okay, so ELISA is enzyme linked immunosorbent assay. So enzymatically color coded or color reaction, which is labeled, and based on intensity, you can find the presence of bacterial antigen. Another technique is latex agglutination is there. This enzymatic technique in that one technique is chair side diagnostic test is there. So evaluate is a test chair side diagnostic kit is there, which is for detection of the detection of basically Actinobacillus actinomycetum commitans, Porphyromonas gingivalis, and Tenerella forsythia. So all these three microorganisms, well is there, a filter paper is there, which is coded with the, or which is lined with this bacterial antigen, and you can find identification. When you take plaque sample and you can put in that well, you will able to, based on the color intensity, pink color intensity is there, and there they have given a what we call it as a scale, visual analog scale. So based on that color intensity, you will find the amount of microorganisms which are present. So that is the chair side diagnostic test based on ELISA technique, that is enzyme linked immunosorbent assay. The chair side diagnostic kit for this is evaluate. Keep in mind, it is there in book. Still, keep that thing in mind. Evaluate is a chair side diagnostic kit. The next is enzymatic technique, which is there is BANA. BANA is a colorless substrate, and basically the three microorganisms, that is Candidella forsythia, Porphyromonas gingivalis, and Treponema. All these three microorganisms, they share a common enzyme, trypsin-like enzyme. So this trypsin, what it does, this colorless substrate, which is there, BANA, benzoyl arginine naphthalamide, this BANA is cleaved. So release of chromophore, that is B naphthalamide, which turn orange red when we add few drops of fast garnet. So this color reaction takes place and color changes to orange red, and based on that you can find these microorganisms are present. So we can, with the help of BANA, BANA is also a chair side detection test. Perioscan is the name for this test. We can detect presence of these microorganisms beside dental chair. Chair side diagnostic test means what? If your patient is sitting in dental chair and immediately you want to diagnose presence of microorganism, you can find these tests useful for detection of this microorganism. But only the disadvantage with chair side diagnostic tests like evaluate, mana is what only you can detect, only few microorganisms can be detected with the help of these technique. Okay, only known microorganism, that is Tenerella forsythia, Porphyromonas gingivalis, Treponema. But the thing is what, you can detect, you cannot detect other periodontal pathogens which are also important, like a Actinobacillus, Actinomycetum commitans. All these are not detected properly. Okay, so these are the chair side diagnostic tests which can be done. Now we'll see some molecular biology techniques, that is diagnostic assay based on molecular biology technique. These, I think you people have gone through these technique in your 12th also. It was there in 12th, right? So molecular diagnostic test or techniques. In that, few techniques are there, like agarose gel electrophoresis is there. Other than hybridization is there. Then along with that, PCR is one technique is there. PCR polymerase chain reaction. Agarose gel electrophoresis is useful for separation of microorganism because based on the molecular weight, that particular microorganism or that particular DNA will migrate towards the electrodes, positive or negative. Usually, they travel towards that electrode, and you can able to find the, you can able to check um, whether the microorganism is present or not. Once you detect the microorganism, you can multiply with the help of polymerase chain reaction. Polymerase chain reaction or PCR is a technique by which amplification is possible. Amplification means what? Increasing the microorganism by n number of times. Amplification. For that purpose, even a single microorganism DNA is extracted and it is multiplied with the help of this PCR technique. Kare Mullis was a scientist who invented this PCR technique. For that, he won Nobel Prize also. So this is very important important thing which has taken place in molecular diagnostic techniques, that PCR, that is polymerase chain reaction. Polymerase chain reaction gives you the identification or microorganism can be amplified with n number of times. So nucleic acid probe or DNA probe which are present like a probe which we use in the oral cavity instrument. In the similar manner, for molecular diagnostic technique, probe is a non-nucleic acid molecule of a DNA. Means DNA is a double-stranded structure, all of you know, and there is specific coding for each DNA. Okay, so a particular specific microorganism artificially synthesized by this DNA technique, or it is identified actually, and a detection was taken place from plaque sample with the help of DNA probes. These are the segment of DNA which is specific to that particular microorganisms. This probe go and attaches to unwinded DNA and this known sequence with the help of known microorganism sequence will able to know that which DNA is there. So particular microorganism probe which we know, all microorganisms probe has been detected by the year 2000, and with the help of this probe we can identify microorganisms also. These probes are of different type, like whole genomic probe, oligonucleotide probe, then RNA 16S RNA probe. So all these probes are complementary to that particular DNA, and it goes and binds, or it goes and fuses with that DNA, unknown DNA. So you will able to know the type or know the name of that particular microorganism. So PCR we have just now discussed. This is double-stranded DNA structure which gets unwinded, and a particular known DNA sequence will go and fuse to this particular part or specific sequence, and you will find with the help of that probe whether the DNA is of particular microorganism or not. So that technique is also known as hybridization. So hybridization, DNA hybridization. On the basis of that, one more chair-side DNA checkerboard test is there, which is developed by Sokransky et al. And Sokransky, what he did, different 40 microorganisms, periodontal microorganisms, can be detected at a single time with the help of plaque sample. Plaque sample can be added upon that filter paper, and on that filter paper, when you add plaque sample, known that particular means quantitatively also up to 10 raised to 4 um, 10 raised to 4 microorganisms which are present quantitatively also can be detected. So this DNA checkerboard is another molecular diagnostic technique or chair side diagnostic technique or tool. With the help of that we can know different 40 different microorganisms, periodontal pathogens at the same time. Okay, so DNA checkerboard is a chair side, another chair side diagnostic test. Then advances in enzymatic test. So these enzymes are host-derived enzymes or tissue breakdown enzymes or other. Means what happens, the microorganism and tissue interaction takes place. And during that tissue interaction,